All right, great. So good morning one more time and welcome all of you to class. Today we're going to start with the topic ecology. I believe you all have this handout with you, or you can download it from the Google Classroom so that you can follow what's going on. Now, when we talk about ecology is the study of the interactions of living things with each other and their physical environment. So we're talking about interactions here, living things with each other and the physical environment, two things. Okay. Now, the living things on the earth may be organized into 4 different levels of ecological organizations. And these are population, community, ecosystem, and then biosphere. Okay. So those are the things I'm going to talk about now. So what is the population? When you take the population, we're talking about all the members of one species in an area. All members of one species in an area. So that's going to look like this. So let's say that you have dogs. There may be cats. And then human beings. Okay. Now, each of these will form a population because members of one species in an area. So you have like a dog population, cat population, human population, for example, right? So this will form the population. I'll make this the population. Okay. Now, if you take the population, right, and put this all of them together, then this will form a community. All right. So community, we say all the members of different interacting species in an area at a particular time. Then when it comes to the ecosystem, we're talking about all members of the community plus the abiotic factors, or what we call the physical factors. So you take the community plus the abiotic or physical factors that influence them. Okay, so this will be, if we take all this, then the bigger subset here, this will be the ecosystem. So all the community plus the abiotic factors. Plus the community. That will be the ecosystem. Okay. So what are some of the abiotic factors or the physical factors? Some of the physical factors will include, so let's take, if we take temperature, it's a physical factor. Okay, the humidity. How humid the environment is. It's a physical factor, all right? If you take something like water, that would all be physical factors. You know, pressure. Okay. So on, they are physical factors. So we put all this together with the community, you've had the ecosystem. Okay. And then the last term is biosphere. Bio and sphere, all right? Bio means something living. So it's the entire region of the earth where living things can be found. So wherever you find living things on the earth forms the biosphere. The biosphere, a part of the earth where living things are found from the biosphere. Okay. So these are definitions that we should know very well. Okay. Now I want to talk about the interactions amongst living organisms. Nutritional interactions, how they feed. Now, all ecosystems must have 3 levels or basic kinds of nutritional interactions, right? 3 types, in order to be stable and to be self-sustaining, right? These nutritional interactions involve the producers, what are the producers, the consumers, and decomposers. So those are the 3 kinds of nutritional interactions that are needed to make an ecosystem stable and self-sustaining. So what are the producers? The producers, or when you say a producer is an organism that is capable of trapping the sun's energy to make glucose, sugar, in the process of photosynthesis, right? In other words, they're able to produce their own food. They manufacture their own food. Those are the producers, right? And they do that by means of photosynthesis, using the sun's energy. Producers. So example of this would be plants and algae. They're examples of producers. They can manufacture their own food using sunlight. Another name for producers that you come across is the word autotrophs. Autotrophs. Auto means self, right? Troph feeding. So like self-feeding organisms, they make their own food. Okay. So that's why they produce their own food from organic substances. Okay. So those are producers. So we should be able to identify producers in diagrams. We'll come to that. Then the second level are the consumers. The consumers. Now the consumers, they depend on and eat other organisms. In other words, they cannot manufacture their own food. They have to depend on other organisms, right? Another name for the consumers is heterotrophs. Heterotrophs. So know the definition: autotrophs, heterotrophs, right? And there are different categories of consumers. We have herbivores. Herbivore, that means they feed on plant materials, and they're only on plants. Carnivore, eat other animals. And then omnivore, eat both plants and animals. Okay, so herbivore, carnivore, omnivore. So example of omnivore would be like human beings. We can eat both plants and animals. Okay. And then the third basic kind of nutritional interaction is the decomposer, the decomposers. And the decomposers, they are a special category of consumers. What they do is that they break down dead organic matter, and they change it to simpler nutrients, which can be recycled in the ecosystem. So their role is to break down the organism when the organism dies, and then they release its organic content back into the environment. An example would be bacteria and fungi. So these will be examples. Okay. So in questions, we should be able to identify producers, consumers, and then the decomposers. But usually the decomposers will not be shown on what we call the food chain or the food web. I'm going to talk about that now, right? So you see the producers, the consumers, but the decomposers will not be shown, even though they are important members of the food chain. So when they ask you the question, which of these important member of food chain which is not shown, then the answer will be decomposers. An example will be like bacteria and fungi and so on. Okay. Right. So this takes us now to the basic concept of the food chain. What is the food chain? So that's what we're going to look at now. Now the food chain is a chain, a single chain showing organism eating by another organism. Like, so the organism eats one organism and it's also being eaten by another one, right? So a chain showing one organism eaten by another, which in turn is eaten up by another organism. And this is how it goes. So it starts with the producers, followed by the primary consumer. So the primary consumer is the first organism. The first organism to feed on the producers. It's not a primary organism. Consumer, sorry, primary consumer. Then the one that feeds on the primary is secondary consumer, and then tertiary, and so on. All right, you can have quaternary at the fourth level, but most of the time, by the time it gets to tertiary, it will end because there will not be enough energy in the food that is left. So producers, primary consumer, secondary consumer, tertiary consumer. Okay. And you have to be careful with the arrow. When they draw diagrams, you have to look at the arrow. The arrow always points towards the in the direction of the organism doing the consumption. All right. So so you can read this as the producer produces food for the consumer, the primary consumer. The primary consumer produces food for the secondary consumer and produces food. You know, forward moving arrows like this. Okay. So you can have the arrow going backwards, let's say from this. If they draw something like with this, the arrow is going like this wrong. Okay, that'll be wrong. That's to go in the direction shown. Okay, point towards the one feeding on the other. So let's take an example of this. Okay. So here you have grass eaten by the grasshopper. Grasshopper is eaten by the toad. Toad is eaten by the snake. Snake is eaten by the hawk. Okay, that's how you're going to read this. Or if you want to read backwards, it means that grasshopper feeds on grass, toad feeds on grasshopper, snake feeds on toad, hawk feeds on snake. Okay. So that is a simple food chain, for the food chain. Okay. So if you have a simple food chain, you can have a question where, let's say the food supply is cut, let's say the toad population goes down. So you have the toad population, if this goes down, so this is down, maybe a disease kills them. What is going to happen to the grasshopper population? All right, so in this case, based on the food chain, the grasshopper population will increase because there's no toad to feed on this, you know. what will happen to the snake population? The snake population will go down because do not have toad to feed on, right? So this is based on the food chain that is given to you. When they give a food chain, don't second guess yourself. Don't be assuming other things. It's, oh, maybe the snake has food somewhere, or the grasshopper something else. Focus on the question. It's possible, but focus on the diagram given to you. So the questions on food chain and food web, food pyramid, they are very simple, very simple. You shouldn't get them wrong at all. You know, if you follow the diagram, focus only on diagram given to you. You don't bring in any extra ideas, you know. If you do that, you get it wrong. Okay. All right, so that's the simple food chain. And then your knowledge should be based on what is given. Okay. All right, so with that, now we're going to talk about the food web, the food web. Okay. But before I go to the food web, I just want to throw in something here about the energies. Now, when we talk about the energy, the plant is going to have 100% energy from the sunlight, right? The energy will 100%. As you move up, right? So you have 10% of the energy for the first level. So it's always 10% as you move higher up of which one. So this will be 100% energy for the plants. Then the grasshopper will be whatever energy the grass has. This is going to be 10% of that energy. Whatever energy that this one has, this is going to take 10% of it, 10% of it, and so on. So the amount of energy decreases as you go higher up. All right, so now let's talk about the food web. Now, most animals are part of more than one food chain and eat more than one kind of food in order to meet their food and energy requirements. These interconnected food chains form what we call the food web, the food web. So if you look at a food web, it's a complex diagram that consists of more than one food chains, right? Because organisms can feed on more than one kind of food. So it looks like a web. So this is a typical example. So let's see how we can read this. I'm going to trace some of the food chains here. So I start with grass. That's a producer, which is the grass. So it is a producer, or the autotroph. Then the grass produces food for the grasshopper. Grasshopper produces food for the toad. Toad produces food for the snake. And snake produces food for the hawk. Okay, so I'm following the arrows, the direction of the arrow, the arrowhead. So that's a food chain here, one food chain. There's another food chain that I see. The other food chain will be from the grass. You go to rabbit, rabbit to snake, snake to hawk. That's another food chain. I see a third one. I see from grass to rabbit, rabbit, food for the hawk. So there are three food chains put together in this diagram to form the food web. Okay. So in other words, the rabbit, if you look at the rabbit, it's producing food for the snake at the same time for the hawk. Okay. Both. If you look at this here. Okay. So the grass is a producer, then the grasshopper, if we follow this food chain along this path. So the grasshopper here would be primary consumer. Primary consumer. The toad along this path food chain here will be the secondary consumer. The snake will be the tertiary consumer, a third level. And then the hawk is going to be what we call a quaternary, fourth level. Quaternary consumer, fourth level of consumption, like primary school, secondary school, tertiary, level, and so on, something like that. Okay. If you go along this path, then the rabbit will be like a secondary consumer here. So this were primary. Rabbits feed on grass on the producer. So this rabbit here will be primary consumer at this level. And then it means the hawk at this level is going to be, if you go along that path, then the hawk here will be the secondary consumer, right? So it depends on which path you're looking at. All right, so you could be asked to identify some of these organisms, which one is tertiary, quaternary, producers, and so on. Okay. Then what's an important member of the food web that is not shown in this diagram? So in that case, it's going to be the decomposers. The decomposers, they're not shown in the diagram. All right. So some other questions that could be asked would be questions like this. What will happen to, let's say, the snake population if the toad population goes down, right? So let's see what's going to happen. So the toad population goes down if this goes down. So let's cross this out. Toad out. What will happen to the snake population over here? If the toad population is down, we still have rabbits. The snake feeds on rabbits here. That's an alternate source of food. So the snake still has a rabbit to feed on. So the snake population, it's unlikely to change. That possible, maybe someone audience is unlikely to be affected. So we still have some source of food to feed on. Now, what about if the grasshopper population goes down? What will happen to the toad population? Grasshopper population is down. If the grasshopper population goes down, that means that now, if you look at it, the toad has no other source of food. So it means that the toad population would decrease, or they'll die off because there's no other food for them based on the diagram. So as I said, your knowledge should be based just on the diagram that you're seeing in front of you. That's the knowledge used to answer the questions. Okay. Which one is a producer? Is a grass, and so on. Then there's another type of diagram known as a food pyramid. The food pyramid. So the food pyramid shows the relationship between the organisms again in the pyramid form, like this. Food pyramid. So the base, which is very broad, the broader part, you have the producers, they form the base, right? And then again, we have the primary consumer, secondary consumer, tertiary, and so on as you go up the pyramid. Okay. So this diagram is like an energy diagram. The energy decreases as you go up, decreasing energy, 10% of the one before it, and so on. Okay. So you can have the same type of questions. What happens if there's a change in population in one of the levels? Okay. All right. Okay, so let's go through what I have here. So this interdependence of population within food chain helps to maintain the balance of plant and animal population within a community. For example, when there are too many grasshoppers, there will be insufficient trees and shrubs for all of them to eat. Many grasshoppers will starve and die. Fewer grasshoppers means more time for the trees and shrubs to grow to maturity and multiply. Fewer grasshoppers also means less food is available for the toads to eat, and some toads will starve to death. When there are fewer toads, the grasshopper population would increase. Yeah, and then I gave a question, predict the effect of low population of snakes in the pyramid, as I discussed. Question, which category of consumer organisms is usually not shown in the food chain or food web, right? So questions like this. Yeah. Okay. Now the next one is to look at some other feeding relationships, long-term feeding relationships, long-term. And this is what we call symbiosis. Symbiosis. Good. It refers to long-term feeding relationship between two or more organisms. It's over a long period of time. Some organisms establish relationships. Now there are 4 types of relationships that fall under symbiosis. The first one is parasitism, then mutualism, second, third, saprophytism, and the fourth, commensalism. Okay, those are the 4 groups of 4 types of symbiosis or symbiotic relationships. Now, what's parasitism? Think of the word parasite, parasitism. So under this type of relationship, one organism feeds on another and causes injury or harm to it. So there's a harm or injury caused. One organism feeds on another and causes injury or harm to it. So in the question, you ask yourself, is the organism causing injury to another organism? If that is the case, then you have a parasitic relationship. Parasitic relationship. Okay. Let's take an example here. Example, like if you take bed bugs feeding on the blood of humans, right? So the bugs are parasites. That's a type of symbiotic relationship. The bugs are causing injury to the human being by feeding on the blood. So that's a parasitic relationship. Tapeworm in the intestines of human beings, same thing, it causes injury. Plasmodium falciparum, it causes malaria. So those are causing injury. So these are parasitic relationships, parasitism. So when I give examples, think about if there's look out for injury, then you can say the relationship parasitism. Now mutualism, mutual, like beneficial, right? So if you think of mutualism, I mean both organisms benefit from the relationship. It's like I benefit, you benefit type of relationship, beneficial. Examples. If you watch, let's say the National Geographic and those science channels and other channels, sometimes you see these organisms, and they show organisms and the relationship that they form with each other. Yeah. It's very interesting when you watch some of these shows. Now let's take example of mutualism here. you have the ospecus. The ospecus, it's a kind of bird, right? So the ospecus, it lands on the rhinos or zebras, and then they eat ticks and other parasites that live on their skin. Now, what's happening here? The ospecus get food, and the rhinos or the zebras are relieved of the pests. So both are benefiting. Food for the ospecus, and then the rhinos are relieved of the ticks, which are pests. So there's a beneficial, both benefits. So that's a mutualistic association. Then another example, we take the spider crab and algae. Spider crabs live in shallow areas of the ocean floor where they are easily preyed upon by other sea creatures, right? They are preyed upon. Now, the greenish brown algae lives on the back of the crabs. So this made them blend in with their environment so that they become unnoticeable to the predators. They both benefit because the algae gets a good place to live and the crabs get camouflaged, right? So that's beneficial relation, both benefits. One gets a good place to live, that's the algae, and then the crabs are also camouflaged. They come from the environment so that they can be easily seen by the predators. Okay. So another example, think of the bee and flower. Bees fly from flower to flower, gathering nectar, which they turn into food. While feeding on the nectar, the bees get some pollen on their hairy bodies. The pollen is transferred to the next flower when the bee lands on it, leading to pollination. So in this mutualistic relationship, the bees get their food and the flowering plants get to reproduce. Right? So that's a beautiful relationship that we see here. Both organisms are benefiting. Mutualism, like mutual fund. You put the money together, you all benefit by putting the money, investing it. Then the third one, the commensalism. Commensal relationship, commensalism. So this is a relationship one organism benefits, but the other does not benefit. So one benefits, the other one does not benefit or get injured. So only one is benefiting. The other one doesn't get anything, or it doesn't get injured too. Example, the shark and the remora fish. Again, if you are somebody who watches science channels on National Geographic, you see when a shark is swimming, this little fish swim alongside the shark. We call them remora fish. What are they doing? The remora fish feeds on the crumbs that fall out of the mouth of the shark. The remora fish benefits from feeding on the crumbs, but the shark does not benefit or lose. Okay, so the shark doesn't lose anything. The food just wastes. That's just waste coming out of the mouth. But the remora fish is benefiting by picking up the pieces or the crumbs. So that's commensalism. Another example, the cattle and cattle egrets. The cattle egrets are birds that live near cattle. When a cattle graze, their movements stir up the insects. So use their feet, then they stir up the insects from the grass. So as the insects fly off, then the birds have their insects, and the cattle are unaffected. So the birds are gaining food by feeding on the insects. The cattle doesn't get anything, from that. Sorry, give me 1 second. All right. Okay. So these are examples of commensalism. Commensalism. Then the next one is saprophytism. Saprophytism. So this is a relationship that involves organisms feeding on non-living things. The organism feeds on non-living things. Okay. So we call the organisms saprophytes. Saprophytes. So example of a saprophyte will be mold. Mold is saprophyte. So mold and bread, this relationship is saprophytism. The mold is a fungus, it's a living organism that feeds on the bread, which is a non-living organism. Okay. So that's saprophytism. Okay. So if you're given examples like this, scenarios like these, you should be able to determine where each of them fall under. Okay. Any questions so far on this? Relationships, feeding relationships, and food chain and food web. Okay. All right. All right, we'll look at another topic, which is mixtures. Mixtures. A mixture consists of two or more substances that have been combined such that each substance retains its own chemical identity. All right. So you're mixing two substances. There's no chemical reaction occurring here, right? So they maintain the identity. Not no chemical combination. It's a physical, so mixture is like a physical change. Now, so that is a combination of substances which are not chemically combined together. Now, the key characteristics of mixtures are: one, they have the same properties as their components. So if you have, let's say, water, sand in water, there's no chemical combination there, right? The water maintains its properties, the sand maintains its properties. Then there's no fixed proportion between the components. Since it is not a chemical change, it can be combined in any ratio. So no fixed proportion. Any amounts can be put together. The components can be separated from the mixture. So you can separate the components. So these are key characteristics of what a mixture is. Yeah, I should be able to separate the water from the sand, for example, right? Because there's no chemical change between the two. So examples, a mixture of sugar and salt. Air is a mixture of gases with nitrogen, oxygen, carbon dioxide, etc. A mixture of flour and sugar, salt and sand mixture, salt and water. Okay, examples of mixtures. What are the types of mixtures? There are three main types of mixtures. You have a solution, you have a suspension, and colloids. Solution, suspension, and colloids. Those are three types of mixtures. Okay. You want to look at the definition for each type. When you take a solution, let's begin by looking at the solution. Solution is a homogeneous mixture. So I'm going to highlight that. When I say homogeneous, what does that mean? It means that there are no boundaries between the components, right? So no boundary is visible in the entire solution. You don't see any boundary. Let's say if I have water and sand, I can see a boundary between the water and the sand. When we have a mixture, you have a solution, you don't see any boundary in the entire solution. Okay, so it's called a homogeneous mixture. So a solution is a homogeneous mixture of a solute in a solvent. Now, as we know, the solvent is what is doing the dissolving. So if you take something like water water is a solvent. And what is being dissolved, what you put in to dissolve, is what is going to be the solute. The solute. So whatever I put in, solute. Let's say salt will be the solute. Put it in. So usually the solvent forms the bulk of the solution. Okay. All right. So the major component is called the solvent, and the minor component is called the solute. The dissolved substance is known as a solute, and the solvent that does, and the substance that does the dissolving is known as a solvent. For example, in a salt solution, the water is a solvent and the salt is solute. The components of the solution may not be separated from the solution by leaving it to stand or by filtration, right? So you can't filter. If you have a true solution or homogeneous solution, you cannot filter to separate the components. I can't use a filter to separate the salt from the water, you know. And if another example like sugar and water, sugar dissolved in water, just like the salt. I can't separate the salt from the sugar from the water by filtration. I have to use a different means to separate the two. Now, degrees of saturation. We'll look at degrees of saturation. A substance can be saturated, it can be unsaturated, or it could be super saturated. Saturated, unsaturated, saturated, or super saturated, right? Now, a solution is unsaturated if it can dissolve more solutes at a given temperature. So let's say that I put salt or sugar in a solvent at room temperature. It's going to dissolve. If I add more, it will keep dissolving, right, at that particular room temperature. So if you keep dissolving more, then we say that it is unsaturated. You can dissolve more. Now the solution becomes saturated if no more solute can be dissolved with the temperature remaining constant. So if I keep adding more of the, let's say the sugar, it will get to a point where it's not going to dissolve again at that particular temperature. So say that it is saturated at that temperature. Now what's going to happen if I warm it up, increase the temperature by heating it up? If I increase the temperature, then I can dissolve more. So in other words, temperature is going to affect how substances dissolve. So temperature affects solubility of substances. In solvents. Okay, so good to note that. So if we change the temperature, you can dissolve more. That's what this means. Okay. So you can keep dissolving more and more and more and more. And it's going to get to a point where it's not going to dissolve again. No matter what you do, if you increase the temperature, keep adding, get temperature, it's not going to dissolve anything again. So at that point, we say that the solution is super saturated. Super saturated. Okay. So a super saturated solution contains more solutes than it will dissolve. It cannot dissolve anymore, right? Where more than to dissolve. If it were in equilibrium, it would be undissolved solid. So if the dissolved solid were in equilibrium with the undissolved solid, right? So you're going to have the particle seeds now appearing in the solution. They cannot dissolve more, supersaturated. Okay. So those are the three types of, or the 3 degrees of saturation. Then we have suspensions. Suspensions. So a suspension is a mixture of liquids with particles of a solid, which may not dissolve in the liquid. All right. The solid may be separated from the liquid by leaving it to stand or by filtration. So the key is a mixture of a liquid with particles of a solid. Example of suspension, let's say if I have like sand in water, you know, or if I take, let's say, some form of powder, I put it in water, you have a suspension. Now the suspension, if we leave it, you see the components will separate. If you shake it, then like you see it mixing up. By leave it for some time, they separate. So some medications are suspensions, especially for the life of the kids. A lot of them are suspensions. That's why they tell you shake it before you use it. Then you have colloids. Colloids. So colloids are homogeneous, non-crystalline substances consist of large molecules or particles of one substance dispersed in a second substance, right? And it's been broken down small particles in another substance, but it's homogeneous. You cannot see any phase between the components, like any boundary, like any boundary between components. Okay. Example of colloids would be like gels, salts, emulsions. And the particles do not settle and they cannot be separated out by ordinary filtration or centrifuging like those in suspension. Right? So you can't use filtration to separate them. The particles are dispersed, but you cannot separate them like suspensions. Okay. So know some examples of colloids. Okay, emulsions, gels. So what are the emulsions? So the emulsions, this is a fine dispersion of minute droplets of one liquid in another. So it's a liquid, fine dispersion of one liquid, droplets of one liquid in another, in which it is not soluble or miscible. They have what we call emulsion. So like liquid within a liquid type of thing. Now that is a colloidal suspension of one liquid in another. Emulsions may be temporary or permanent. Temporary ones separate when left to stand for some time. Now, examples of emulsion, if we take oil and vinegar, put oil and vinegar, it's a temporary emulsion. Mayonnaise. Mayonnaise is egg yolk in oil. The egg yolk in oil, that's mayonnaise. Okay. So the egg yolk dispersed in the oil. That one is a permanent emulsion. Okay. So mayonnaise is an example of an emulsion. It's a colloid emulsion. One liquid dispersed in another. Then the soil, these are colloidal substances of small solid particles. So here we are looking at small solid particles in a continuous liquid medium. So you have small particles dispersed in the liquid medium. Example would be blood. We take a blood, ink, paints. They are all examples of soils. Small solid particles dispersed in a liquid medium. If the dispersion medium is water, the colloid is referred to as hydrosol. So when we say hydrosol, means water is the dispersion medium, the one that is the one in which the solid particles are dispersed. And if it is air, then we call it aerosol. Aerosol. Okay. So if you think of the spray, the things that we spray, right? Those aerosols. They have small solid particles dispersed in air. Aerosol. Then the gels. So the gels are colloids. They are colloids in a more solid form. They are more solid form than the soil. Right. So there's particles again that dissolve in a liquid, but then it's more solid than the soil. So the dispersed phase has combined with the dispersion medium to produce a semi-solid material. An example would be like jelly. You take jelly, it's an example of gel. It's more semi-solid. All right, so if they give examples, you should be able to classify them as emulsion, sol, gel, and so on. Okay. Okay. All right. Does anybody have any question on these ones? Okay. All right, so let's move on to another topic, which is the electroscope. Electroscope. Want to look at the electroscope. An electroscope is used to detect the presence of static electricity. That's all. It detects the presence of static electricity. So in other words, if you want to find whether there are charges on a certain material, you use an electroscope to determine that. It consists of two metal leaves. Example, gold leaves. So this is a diagram, a simple diagram of how the electroscope looks like. So you have metal leaves. This is a jar. Because this is a jar here. And they have the metal leaves here. The one labeled A will be the metal leaves. And it can be made of gold, for example. Suspended from a metal hook. So the B is a hook. C is the jar, let's say glass jar. So it's a very simple setup. And then D is going to be like a plate, metal plate. Okay. So how do you use it to detect charges? If a negatively charged body is brought near the terminal of the electroscope, so if I bring a negatively charged body here, an object that's negatively charged, for example, if you comb your hair the comb gets charged, you know. So you can use the electroscope to detect the type of charges on the comb. Okay. So let's say that you have a negative charge on it, and you bring it to the terminal of the electroscope, the plate. It will cause electrons to repel to the metal leaves. So remember, opposite charges attract. So these negative charges will attract positive charges from the rod to the plate. So you're going to have positive charges here. And what's going to happen? All the negative charges will be repelled to the end of the rod. So it's going to go to the leaves. So negative charges will be repelled to the end of the rod because they are opposite charges. Now, what's going to happen to the leaves here? Since the leaf on the left side is negatively charged, the right leaf is also negatively charged. Same charges. So it means that the leaves will spread out, they'll spread apart. They will spread apart. And that will tell you that there's a charge on the material that you brought close to the plate. Same thing will happen. Let's do the opposite. If I take something that is positively charged, now this is going to attract negative charges from the rod to the end plate here. So you're going to have negative charges attracted. And all the positive charges will be repelled to the end of the plate. Sorry, the leaves. So you have positive charges repelled to the end. So the leaves will be positive. Positive charge on both leaves. So since they are same charges, the leaves will move apart. Okay, they move apart. And I'll tell you that there's a charge on the object. So the amount the leaves open is proportional to the charge of the source. In other words, the more charges you have on the object that you brought close to the plate, the wider the leaves spread out, and so on. So that's why it's proportional. The amount they open up is proportional to the charge of the source. Okay. So it is just a simple setup to detect charges. Okay. All right. Now let's look at waves, overview of waves. There are different types of waves. We have mechanical wave and electromagnetic waves. Mechanical and electromagnetic waves. A mechanical wave requires a medium for its transmission, whereas electromagnetic waves does not. So you get know the definitions here. So two types of waves, mechanical and electromagnetic. Mechanical needs a medium for its propagation. Electromagnetic wave does not need a medium. Let's take an example. If you take sound waves are mechanical waves. That is, sound does not travel in vacuum. So if I take all the air around me and I'm in vacuum, if I speak, you not hear me because there's no air for transmission of the sound wave. So sound needs a medium like air. Or if I tap on the table, the wood, is a medium. The sound can travel through the wood. So the wood is a medium. Yeah. If you take something like light waves are electromagnetic waves. Light waves are electromagnetic waves. Because they are electromagnetic waves, they don't need a medium for their propagation or their transmission. Right? In other words, light can travel in vacuum. Right? So we can see the sunlight, whether there's air, there's no air, you can still see the sunlight. You can feel the radiation of the sun, the heat from the sun. It doesn't need a medium. They are electromagnetic waves, like UV lights, all those things, microwave, they're all electromagnetic waves. They don't need any medium to travel through. Okay. Now, all the waves share some common properties, which we're going to look at. And that's how they look. If you plot the graph of the distance from the origin, let's say you have this X and Y axis. And the origin, let's call it O, the starting point. So this is time. So we're talking about time on. And then the y-axis will be the distance, the vertical displacement from the origin. How far they're traveling from the origin, distance from origin. Okay. It always takes this form, sinusoidal graph, like this. And keeps going. Okay. The sine wave. Okay. There are some properties that we want to talk about. And the first one is the wave length. What is a wavelength? Wavelength. You should be able to identify all these things. Usually give a diagram like what I have here, and then you identify the different parts. So what is the wavelength? Let's take the pendulum. If I take the pendulum, there's a starting point here. It's hanging, and then I move it up. Displace it a little bit. It's going to swing to and fro, right? So when I move it from the original, this is a starting point, and I move it up here, I've displaced it from its origin. So the distance, which is the height to which you displace this, the maximum distance is what we call the amplitude. The amplitude is the maximum displacement from the original position. So this is a reference line. So the maximum distance is here. That would be the amplitude. Okay. So this is the amplitude. Maximum displacement from the resting position. I can also have another amplitude. Below here is also an amplitude. It's also maximum displacement. This original line is the reference line. From here there, okay. So this is an amplitude, another amplitude. So if I come here, this is an amplitude here, that's amplitude. This is an amplitude. These are our amplitudes. Okay. Now let me label some parts of the wave, and I will continue with the definitions. Now the maximum, the peak, the one above, like above the reference line, is known as a crest. And then the maximum displacement below the reference line here, this is called the trough. like the valleys. The mountains are the crests, the valleys are the troughs. Okay. So the wave is made up of crests and troughs. Okay. So that's amplitude. Now, what is the wavelength? The wavelength is going to be the length of one complete cycle. How long does it take to complete one cycle? Right, time taken to complete one cycle is what we call the wavelength. So if you start with a pendulum, you move it up, it swings down. So this is going to represent it, go like this, up, maximum displacement, which is here. So that takes you here. Then you're going to swing back. That brings you down back to the origin. And then moves again to maximum position. So that this brings you down to the trough. And then swings back to the origin. So that brings you here. That's how it goes. Okay. So all this here, this is one cycle, one complete cycle. One complete thing. Down, the complete cycle. So the time taken to cover one's complete cycle is what we call the wavelength. So from here, this point, all the way to this point is the wavelength. Okay. The time to complete one cycle, that's the wavelength. I can identify more wavelengths here on the same graph. Let's increase this a little bit. This is a crest. So if it starts from the crest, so let's say here, and then swings back and back, that's another wavelength. So from crest to crest, it's also a wavelength. It's a complete cycle wavelength. Okay. From trough to trough, this trough here, to another trough here. That's also a wavelength. Okay. It's a complete cycle. So I go from here down, back, complete cycle. If I start from here, go up, down, and back, that's a complete cycle. So the distance or the length, the time taken to complete that cycle is the wavelength. Okay. So show it to different wavelengths in diagrams. Okay. So when I come here, what are the wavelengths in this diagram? I can say that this is one wavelength, A to A all the way to E. It's a wavelength. Okay. If B to F, that's a wavelength. I can identify another wavelength if I start from C to G. C to G is also going to be a wavelength. C to, that's a wavelength. It's a complete cycle. Okay. Then we have frequency. The frequency is the number of cycles per second. How many cycles you have in 1 second, complete cycle. That is what we call a frequency. Frequency. So if I take a wave, let's say it is 1 second, from 0 to let's say 1 second. How many cycles do I have in a second? I can have something like this. It means 2 cycles per second. Or I could have so many cycles per second. I could, sorry. I could have this. Let's say something like this. So I have so many cycles per second, but let's say 1 second, right? So now we look at how fast the wave is traveling, it's completing the cycles. Okay. So the number of cycles per second is a frequency. High frequency means complete the cycle faster. Low frequency is taking longer time to complete the cycle, right? So now we're going to relate this to hearing. How is it related to hearing? Frequency determines the pitch of sound. So we see frequency, think about pitch of sound. So some people have low pitch sound, some people have like, ooh, some, ooh, so on, like high pitch, some people can even sing to break glass of water or just a glass that is on the table. Go to have high pitch sound. They can make a high pitch sound that can break things. So think about that in terms of hearing. Okay. Now amplitude. How is amplitude related to hearing? Amplitude has to do with the loudness of sound, loudness or intensity of sound, right? So the size of the amplitude is related to the loudness. So the large amplitude, that means loud sound. Low amplitude, low sound. So that comes out in questions too. Okay. So what we're saying is that the frequency when it's low, think about low pitch of sound. When it's high, high pitch sound. Whereas amplitude, low, it means sound is not loud. High means loud sound, and so on. Low sound, high, loud sound. Okay. Right. So this is an example of a diagram that you could get. And you have to determine what you see. Okay. So someone should try this one. Okay. So the wavelength of the wave in the diagram above is given by which letter? So which one will represent the wavelength? Someone can try that one. Which one represents the wavelength in the diagram?
A.
And could you say that again?
A.
A, correct. Yeah. Got crest to crest, so it's A. All right. The amplitude of the wavelength. Which letter? Amplitude.
B.
Try again.
D?
D, correct. It's D. It's D. If you look at B, B is made up of two amplitudes all the way here. So that is double the amplitude. Okay. The amplitude has to be the maximum difference. This is a reference position. The line, dotted line is a reference position. So this is amplitude. Yeah. And radio waves are what type of waves? Radio waves doesn't need a medium. Radio waves that don't need a medium. So because I need a medium, it has to be electromagnetic wave. So this is like a diagram one that they could get. Okay, great. Okay. All right, now let's look at enzymes and catalysts. Enzymes and catalysts. All right, so what are catalysts? Now, catalyst is any substance that can speed up the rate of chemical reactions. So they speed up the rate of chemical reactions. So those are catalysts. Now note that the catalyst does not take part in the reaction. They do not take part in the reaction. All that they do is that they just speed up. They are there to speed up. So at the end of the reaction, they remain unchanged. They remain unchanged at the end of reactions. They just speed up the rate of chemical reactions. Example, if you take enzymes, enzymes are catalysts. So refer to enzymes as biological catalysts because we find enzymes in living things, right? If it's not living, then just going to be like a catalyst, right? If it's biological, if we find it in living things, then we call it enzymes. So enzymes are proteins that speed up the rate of chemical reactions in living organisms. And one of the properties of enzymes is that they are specific. They act on specific substances. Enzymes act on specific substances and they are proteins. Okay. So because they are proteins, they can be affected by temperature. They can be affected by pH, because those things can change the structure of the protein. So enzymes function well within what we call certain optimum temperature and pH ranges. If you go outside those range, then either they don't work well or they are destroyed, or they become what's called, we say they are denatured. Their nature changes. You destroy the nature of the enzyme. All right. So if I take an enzyme, let's say you have a protein that looks like this. The enzyme has certain places where the substance that it's going to act on binds. So let's say that this is the site that the substance is going to bind. We call this site the active site. Active site, the site where the substance acts. And the substance that is going to go here usually should fit perfectly. So it should have a perfect fit. And that substance is what we call the substrate. The substrate. Okay. Has to be like a perfect fit. So there's something called a lock and key mechanism. Like for every lock, there's a particular key that can fit in, you know. So once the substrate binds, they have what we call the enzyme substrate complex, right? Then the enzyme can now help in breaking down the substance. Okay. The enzyme may also have other sites on it where the substrate doesn't bind, and that those are called we call them allosteric sites. Allosteric sites. Okay. So those are the substrate doesn't bind to those sites, but other things can bind there to either change the shape of the enzyme or even to enhance its action, right? So some of the substances can bind here, they can inhibit the enzyme. So we call them inhibiting substances. They can inhibit. The enzyme. Okay. All right. So let's draw the energy, the diagram for the graph for pH and then temperature. How does this look like? When it comes to the pH and temperature, the graph looks like this. It has the bell shape if there's enzyme activity. You have a bell-shaped graph like this. Now the enzyme will always work best, as we said, in optimal pH. So the best activity is that peak is somewhere here, right? And this will call the optimal pH. If you go outside that, it's not going to work too well. So always the narrow range. If I put another, let's say it's going to work within a narrow range of pH around the optimum, somewhere in between. It's going to work well in that region. If you go below, then it's going to drop as you go further away from. The same with temperature. If you take temperature, it follows the same thing. If I put temperature here, it follows the same pattern. So this will be your optimum temperature, the best temperature that will work. Very narrow range. Okay. So if you take like a living source like human beings, it works well within a temperature of 37 degrees Celsius. You know, enzymes that work well in that range, within that temperature range of within a human being. So in the lab, if you're going to use enzyme in the lab, you have to use a temperature around 37 degrees Celsius for that enzyme must work well. Okay. So this is a quick overview of what enzymes do. Yeah. So if you have a reaction, let's say A plus B, then they're going to put the enzyme somewhere here. The enzyme will be here. And then give me you maybe C. Yeah. What will happen to the enzyme at the end of the reaction? It's going to be the same. It doesn't change. Yeah. It's going to be the same. So what happens is that, so if let's say I have A plus B, so let's say that I have B, I want to change it to C. To a substance C. Now the enzyme is going to form B, is going to form what you call the enzyme. It's going to bind to the enzyme to form the enzyme substrate complex. So B with the enzyme form the enzyme substrate complex. Okay. Then the product will be formed. And once the product forms, C will form, then the enzyme is released. You have the enzyme back. So that's how it looks like. Okay. I'm going to draw an energy diagram to show you how the enzyme works. How the enzyme works. Now, most chemical reactions have low energy level, right? The reactants, so let's say we have a reactant here, A plus B. Okay. Usually they have low energy levels. So because of that, it's going to take a long time before they can react. You know, reaction involves the electrons, they bombard against each other, you know, the electrons are vibrating. So, excuse me, when they have enough energy, they combine to form reaction. But since they don't have enough energy, enzymes comes in to help. So under normal circumstances, without the enzyme, this energy here, let me put energy. There's a minimum energy that every reaction has to overcome before they can change to products. So let me just put reactants here to make our life easy. The reactants and then products. It's almost like a hill. They have to have energy, like the energy to push up the hill. So once they get up the hill, this where has to get to this here, this level, energy level. Once they have enough energy level, then they can now form products. They break down to products like this. This is what we call the activation energy. From the reactants here and the energy, this to the peak energy before reaction can occur, is called the activation energy. The minimum energy that they need, the molecules, the electrons need before they can combine to form a reaction. It's called activation energy. Okay. Now, what do the enzymes do? Enzymes come in to lower. So instead of the molecule getting all over all the way to this level, the enzyme will come to lower the activation energy. So it's going to lower the hill to something like this. Okay. So now it's going faster because you don't have to take all the way up there. Enzyme lowers the activation energy. Okay. So it's faster. That's why it's faster. So this will be with enzyme, the green one, when enzyme is present. We lowers it. Okay. So when you see a question that says that enzymes lower the activation energy, that is true. That's what it does. It lowers the activation energy so that the reaction can proceed faster. Okay. So I believe this should be enough for answering questions involving enzymes for the test. Okay. All right, so now let's look at the gas laws. Gas laws. The gas laws. There are few laws that govern how gases behave. And the first law is known as Boyle's law. Boyle's law. Know this also very well, and it's scenarios. I'm going to give you some scenarios based on these things. So let's look at Boyle's law by Boyle. Boyle's law states that at a constant temperature, the pressure of a gas varies inversely as the volume. So we have what we call inverse variation, and that's called a Boyle's law. At a constant temperature, so note the pressure is constant. The temperature is constant here, very important to note. At a constant temperature, the pressure of a gas varies inversely. So talk about inverse proportionality as the volume. So in math, when you talk about inverse proportionality or inverse relationship, it means that as one quantity goes up, the other quantity goes down, you know, almost like having like a negative slope. One goes up, one goes down, inverse relationship. So if you draw the graph, like goes, the pressure versus volume, inverse relationship. So as volume is increasing, you see that the pressure is going down. As volume decreases, pressure goes up. So to put it in the diagrammatic form, then we say that pressure up, volume down. So that's one. If pressure goes down, your volume goes up. That's the summary of the law. At a constant temperature, you're not changing the temperature, maintain the temperature. Right. So here you can think of a diagram like this, where you have, let's say, the piston and the plunger. or you can think of the pump, the type of, like for bicycle. It is that volume and pressure is what you apply here. Let's call the volume pressure 1, volume 1. If you increase the pressure, that means you push it down, the plunger down, or the pump handle down. Pressure 2 up, that means look at small volume, the volume has decreased. Okay, small volume compared to this. So the pressure up, volume down. So that's how I can remember the relationship. The Boss law. Okay. So mathematically, if you put in formula form, if you multiply the initial pressure, this over here, as a formula. So P1 V1 is equal to P2 V2. Okay. If you multiply the pressure with the volume, it gives you the same as the final volume times the final pressure. It's a constant value. That's what that means. Okay. Then we have Charles' law. Charles' law talks about the relationship between volume and temperature, whilst keeping the pressure constant. Okay, pressure constant. So that is known as Charles' law. So it says that, so Charles' law, at a constant pressure, the volume of a gas varies. Here we talk about direct variation, directly as the temperature. So direct variation. Direct variation, that means that as one goes up, the other one also goes up. They all increase in the same direction. So like going like a positive slope. Okay. They increase in the same direction. Okay. So to put it in a diagram form, V1, sorry, so it means pressure, volume up, temperature up. Okay, that's what temperature up, volume goes up. If the volume down, it means so temperature down, volume down. Same direction of change. That is Charles' law, with the pressure being constant. So a typical example that you can visualize here would be like if you take the balloon. If you increase the temperature, what happens? It's going to expand, expand, expand. It gets bigger and bigger and bigger. If you increase temperature, the volume goes up. If you decrease the pressure, the temperature, the volume would go down, and so on. That's Charles' law. Charles' law. And then this is a formula. If you want to think of the formula, if you divide volume by the temperature, you have a constant. So that one involves division, because the change is the same direction of change. Okay. Then the third law is known as Gay-Lussac's law. Gay-Lussac's law. Now, Gay-Lussac's law says that at a constant volume, now we're going to talk about constant volume. Constant. So Gay-Lussac. This time we keep the volume constant. Now, if we keep the volume constant, it says that the pressure of the gas, so talking about pressure here, the volume constant, at a constant volume, the pressure of a gas varies directly as the temperature. It says again, that's a direct variation. Okay, so in other words, if the pressure goes up or the temperature goes up, pressure is up. If the temperature goes down, pressure is down. Okay, it's a direct variation. Okay, pressure, temperature. Okay, so here you can think about if you are boiling water, put water heat, and then you cover with a lid. What happens? Now, as you warm this up, the water, the molecules escape. The water molecules escape into the air. So you have vapor over here. All right. So what you have vapor in here. So the vapor, as you increase temperature, you see the pressure increases. So that's why the lid gets pushed up. Because the temperature increases the pressure of the gas molecules here. So you say pushes up the lid. So when coming up to a formula, is similar to Charles' law formula. So P1 over T1 is equal to P2 over T2. You know, you just change the volume. And then we have what called a combined. The combined gas law combines all the three laws together. You put all this together, then you get what called a combined gas law. That's this gas law here. You know, so here you can see that you have the P1, P over P1, P direct variation, V over T, direct variation, and then the PV between P and V, they are inverse relationship. Just combining all these three laws into one law. You see the combined gas law. Okay. And then we have the ideal gas law, where it's a PV is equal to nRT. That's the formula for ideal gas law. But there's no ideal gas, you know, but if a gas behaves ideally, then it follows this law. N is the number of molecules. Sorry, as a number of the mole. When you're talking about the ideal gas law, this N is known as this is the number of moles. Number of moles of the gas. Then R is a constant. We call it R is called a gas constant. You know, it's around 8.3 something. Gas constant. So if you get a formula like this, they ask what's N in number of moles, R is a gas constant. Okay. I call it ideal gas constant. Okay, I have it right here, right? Ideal gas constant. Right, so know the three gas laws mainly, the Boyle, Charles, and Lussac. Those are common. And scenarios that I've given. One other scenario that I'm going to give you is respiration, like external respiration. For me to take in air or oxygen, right? The rib cage has to move out. Now the diaphragm has to go down. So rib cage moves outward, diaphragm moves downward. So what has happened to the thoracic cavity? The volume in the thoracic cavity increases. Once the volume in the thoracic cavity increases, what happens to the pressure? The pressure goes down, because that's Bohr's law. Volume increased, the pressure will be down. Now, compared to the pressure outside, the atmospheric pressure is now higher than the intrathoracic pressure. So what happens? Now air will rush in, oxygen will go in. Likewise, if I want to get rid of oxygen, expiration, the diaphragm moves up, it contracts and move up. What happens to the ribcage? The ribcage moves down. So ribcage down, inward, diaphragm upward. So the volume, thoracic volume decreases. Volume down, what happens to the pressure? The pressure will increase. So now I have higher pressure in the thoracic cavity than outside. It becomes higher than outside. So that pushes the gas out, the carbon dioxide out. Okay, so that's a scenario where Bohr's law is operating. Bohr's law. So think about scenarios like that, in addition to all the examples I've given you. Okay, any questions on the gas laws? Okay. All right, so slowly getting to the end, gradually. When we get to this topic, then I'm getting to the end. All right, radioactivity. Let's look at radioactive substances. So we'll talk about radioactive substance, or let's talk about radioactivity first. Radioactivity is a random process that happens naturally as the isotopes in a particular element decays or decay, right? So remember isotopes. Isotopes are elements that have the same atomic numbers, but different mass numbers, right? Those are isotopes. So for example, if I take carbon, carbon 12, atomic number of 6, and the same carbon 6, but this time 14. These are isotopes of each other, right? Isotopes. You know, there's oxygen, that is 18, atomic number 8, and oxygen 16, same atomic numbers, different masses. These are isotopes of each other. Okay. So radioactivity is a random process where isotopes, they break down. Decay here, where decay means to break down, they decay. In other words, they disintegrate. Use the word decay. They break down. So isotopes continue to break down over time. The length of time that it takes for half of the nuclei in the element to decay is called half-life. So I'm going to circle that half-life. We have to know half-life. Okay. So it's time taken for half of the substance to break down. Half-life. So if you come across T half, that's what it means. T half. The time taken for half of the substance to break down. To decay or break down. T half. So there are some nuclei, some elements, their nuclei are not stable. Okay, they are large and they want to break down into simple, stable ones, like uranium. Uranium is a typical example of a radioactive substance. You know, it breaks down to produce other smaller elements. Now I'm going to give you an example of how T life works, because that also comes out in questions. When we start looking at the questions, we'll see some examples. But I'm going to make up a problem now. Let's say that I tell you that a substance has a half-life. Let's say 100 grams of a substance, grams of something. And then this has a half-life of, let's half-life of maybe, let's say 5 days. I tell you I have half-life of 5 days. How much of this will be left? Maybe in, let's make it 15 days. In 15 days, how much of this will be left? You can use this approach I'm going to use. Very simple approach. So we're going to work to 15 days. So it means that every 5 days, half of this will be left. So T half, 5 days. So 5 days, it means that I'll have half of this left. So which will be 50 grams. Another 5 days, half of this will be left. So I have 25 grams. So again, this is 5 days. Another 5 days, half of this will be left. So that'll be 12.5 grams. That's another 5 days. So every 5 days, half will be left. That's what that means, T half. So if I'm doing for 15 days, then it means that I have 3 half-lives. 1, 2, 3. This gives me the 15, all these 15 days. So in 15 days, I'll be left with 12.5 grams. Okay. When you get a question, you can do it like this. Then you get the answer right. Okay. When you're doing chemistry proper, there's a formula that you can use to do this as well. But you can use simple arithmetic like this to work it out with the question that you get. Okay. Half-life, every half-life, half of the substance left, and so on. So they give you 2 half-life, 10 days, like this one, then you stop at 2. 3 half-lives, then 15, and so on. You keep going till you get what you want. Sometimes you can work backwards. Some questions can give you, let's say, after 15 days, half of, let's say you have 12 grams left, 12.5 grams left. So they'll give you this one. Then they ask you, maybe take 15 days to get here. So what will be the original amount in the starting amount? So it means you're going backwards to get this. So if you're going backwards, then you just do times, you multiply this by 2 to until you get what you want. You get to a final. So there are 3 half-lives, then you're going to break it down. Start from this, go first half-life, second half-life, third half-life. Okay. So if there are 3, then you divide the total half-lives, the total days by the number of times you have to get to original. You know. Use a simple diagram like this to work it out. Okay. So once we see a question, we'll apply this concept again. Okay. All right. So that's half-life introduction. And then this will be different examples of different type of decays. Okay, types of what we call radioactive substances types. or decay. You have what we call alpha decay, alpha decay. And this is alpha, like this symbol, that's what we have here. And alpha decay means the substance emits an alpha particle, right? It gives off an alpha particle. It's a type of radiation. So a type of reactive decay or radiations. Alpha radiation. It's alpha particles, a radiation, example. And basically, it's a helium atom, right? Helium has atomic number of 2, and then so it's like a helium atom. That's what alpha particle. So if it starts with a substance that gives off, let's say A, A is atomic number, A is atomic mass, and Z is atomic number. So let's say we have an element X, and it breaks down into an alpha particle by releasing alpha particle, right, to form a new substance. Then you have alpha decay. So we see this, it means we're giving off an alpha particle. Okay. Now I'm going to give you an example very soon, how you can work problems out like we have here. You see A minus 4, Z minus 2. If what happens is that if you add the atomic numbers on the left side, it should be the same as the atomic number on the right side. If I add for the masses on the left side, it should be the same as the right side, and so on. Right? So that's the concept being applied here. Okay. Then you have beta decay. Beta decay means it gives off a beta particle. So beta decay, it gives us a beta particle is given off. Okay. And what's a beta particle? This is a symbol, like alpha beta, right? Symbol. So that's beta. Beta has atomic number of -1, and then the mass is 0. So basically, this is like an electron. Beta particle is like an electron. And then you have what we call I'm going to talk the common ones first. And then have what we call the gamma radiation, the gamma particle or radiation. And this is a gamma symbol, like this gamma, right? Atomic number 0, 0, mass is 0, atomic number is 0. So the gamma particle is basically like a neutron. It looks like a neutron. Yeah. So if you can remember these ones, alpha particle, which is alpha particle, beta particle, gamma particle, those are the common ones. We call them radioactive particles, alpha, beta, gamma. Okay. And then the other ones, if you know them, we have positron, then there is a positive beta particle. Then they have electron, what we call electron capture, where the particle, the element captures an electron. So this captures an electron and becomes a new substance and gives off like X-rays. It gives off X-ray. Okay. Then here we have what we call fusion, spontaneous fusion, and I'll talk about fission, fusion and fission. So for fission, when I say fission, nuclear fission, so types of radiations, nuclear fission, and they have nuclear fusion. Right? Fission means that you are joining the nuclei. Okay, like what we have here, there's joining of nuclei. No, this one is fission. So let me make it simple here. Joining nuclei. So let's say you have A plus B joining to give you a bigger one, let's say C, bigger element C. They have nuclear fission. Nuclear fusion means, no, sorry, I switched the two up. I switch out the two. This is fusion. That's joining, fuse. And then this one I'm going to talk about is fission. So fission is splitting the nuclei. Splitting nuclei. Okay. So let's say I have a large nuclei that breaks down to smaller ones, and then they release a lot of energy, right? And that's what is being shown here. Like that atomic bomb and other things, that's how they created them. Generates a lot of energy. Those small nuclei that we see, if you split them apart, a lot of energy. And some poles are used for generating electricity, like nuclear reactions, you know, and other poles used for bad things. Okay. All right. So these are the properties of the different radiations. If you take gamma radiation, you can use paper to stop it. Okay, you can stop using paper, so it's not too powerful. The energy is not as high as if you compare to beta. Beta is the next one. Beta radiation, you can stop it with metal sheets, like aluminum metal sheet will stop beta radiation. Gamma radiation, the next powerful one. Gamma radiations and like the X and then X-rays. You can stop them using thick lead, lead or steel plates. You can stop them. Then the neutron rays, what we call the neutron rays, that one, you can stop them using like water, tank of water or like concrete, then that can stop them. And I spoke about the types of radiation, like the examples, alpha decay. These are typical examples. So if you take uranium, so uranium is 238 and then 92. If this breaks down, we have it forms this thorium 94 to helium. So this is alpha particle, right? The helium is an alpha particle. As you can see here, it's an alpha particle. And then you have beta radiation, these are examples. Beta radiation, electrons are given off. Electron capture means that thing captures electron, like this. And this is an example of the fission. the fission. This is an example here. Splitting. Now, one type of question that you could get will look like this. I can get rid of this here and get rid of this there. And then put X and Y here. And I can ask you to find X and Y. It's very easy to do this. You compare the atomic numbers and compare the masses. So if you compare the atomic numbers now with 92 on the left side, then we're going to compare to this and that. The atomic numbers on the right side. In other words, 92 should be the same as Y plus 2. The numbers should add up. The balancing equation. So left side should go right side. And then you can solve for your Y. Likewise, 238 should be equal to, now look at atomic masses, the mass. They should also match. So X plus 4 should be equal to 238. All right. So if we do that, that means that here we'll have your Y is equal to 90. And then here we have our X to be equal to 234. You know. So that's how you can do this type of question if something's missing. All right, so for radioactivity or radioactive decay, you know that particles, alpha, beta, gamma, common ones, and then maybe you can add in X-rays. Then nuclear fusion, nuclear fission, and how to use the formula, find missing parts of a formula, like what I've given. All right, very simple questions like that. Yeah. And maybe the properties, as I said, what can stop what, and it should be fine. Great. Then the last, I believe that's the last topic for the science, and then we'll have covered all the things that I need to talk about. All right, so the last one will be light, properties of light. So these are all physics, questions. Properties of light. The first property of light, we've already said that light is an electromagnetic wave. It has certain properties. So the first property we want to talk about is known as reflection of light. Now, reflection occurs at the surface of a mirror. So if you have a mirror and I shine light, so this is light source here, the ray from that light source is known as the incident ray. So this is the incident ray, right? Now, when it hits the mirror, it's reflected in the opposite direction. So it comes in this direction, then it's reflected back in the opposite direction like this. Now, this ray here is known as a reflected ray. So that's a reflected ray. Okay. If I draw a line perpendicular to the surface of the mirror, we call that the normal. So the normal is the line perpendicular. So this is a normal. It's perpendicular to the surface. Okay, it's called the normal. Okay. Now, if I take the angle between the incident ray and then the normal, this is known as the angle of incidence. And then if I take the angle on this side, which is between the normal and the reflected ray, it's called the angle of reflection. Okay. Now, the angle of reflection should be equal to the angle of incidence. The two are equal, right? So keep that in mind, they are equal. So sometimes you draw a diagram, we'll see a question once we go to a book. They draw, maybe this is 30 incident ray, reflected ray. Then they ask you what's this angle here? Let's go R. They should be said 30 because they're equal. Yeah. Sometimes they draw different angles. They can draw one that is maybe this, they make this 30, draw another one, then they'll say 40, another one 50, and then ask you which one, A, B, C, which one it's the reflected ray, and so on. Okay. So reflection occurs at the surface of a mirror. Then we have diffraction. Diffraction. Diffraction is defined as the bending of light waves around obstacles in its path. Bending of light around small objects. That's what this bending. So even though light travels in a straight line, right, it can bend around small objects. And that's what we call diffraction of light, bending around small objects. So when a light wave passes through a barrier with a small opening, it acts as a single point source from where light emerges and spreads in all directions. So in this example here, it's sort of like if you have a slit, a small opening, and you have light comes in, the light doesn't go straight in. It ends up bending a little bit. Like it bends a little bit, not too much, but it bends a little bit around. But this is more, you get a better explanation if you think of the microscope. But a microscope, and I have a bacteria. Let's say this is a bacteria on the slide. Small bacteria. As the light shines on it, it doesn't go through the bacteria. This is like an obstacle, right? So what happens? It bends a little bit around it and comes, you know. So this slight bending around the object is what called a diffraction of light. So under the microscope, you see that there'll be a little bit halo around the bacteria, the particle, like light halo around it, it's bending around it. That's why you see the dots, but you see the bacteria's dots on the slide, you know, dark slots and dots. The light graces the edge of it, diffraction. And then we have refraction of light. Refraction of light is also has to do with bending of light, but this time it is traveling from one medium to another, right? So like from air to water or air to glass. Okay, so refraction will occur when the densities of the objects are different, you know. So if you take this here, let's say this water, right? So you have the fish under the water, let's say in a pond. Now the light coming from the fish, as it hits the surface of water, it ends up, instead of going straight all the way, straight like that, now we can see that it's bending. That's, the person is saying the light bends at the surface because you have air here. Air is less dense than the water. So less dense or has low density and is higher density. Okay. So because the light, the ray is traveling from the high density to the low density, it ends up bending away. If I can draw a normal here, if I draw a normal, let me use a different color. If I draw a normal here like that, you know, the light bends away from the normal as it moves into a less dense medium. When it bends away from the, moves away from the normal, it means that it's traveling faster. If it moves closer to the normal, it means that it's traveling slower. Okay, that's what it means. Fast if it's away from the normal. So this is a normal line. Okay. So because of the bending, it appears as if this ray is coming all the way from this end, from this fish over here. If you trace it backwards, you know. So it makes the fish appear as if it's closer to the surface than it is, you know. So refraction explains why when you look in a pond it appears as if it's shallow, but meanwhile it's deep. You know, you'll get a false impression of how deep it is. Now let me redraw this diagram in another form and label all the parts for you. So in the lab, you can do this using what we call a glass block. The glass block. If you take the glass block and draw a normal, let's say I have a normal here. I'm going to draw some normal here. If I shine a light on this end, it's going to hit the normal like that. Then it's going to bend. Since you have air here, air on this side, less dense. So because it's moving to a dense glass, it's going to bend. The ray is going to bend towards the normal. It's going to bend towards the normal. So you have, looks like that. And then as it comes out of the glass into the air, the light is going to move away from now. It travels faster now. So it moves away from normal like this. So the eye, your eye here, this is going to appear. So this is the normal. So this ray from the light source is going to be the incident ray. So again, this is the incident. Incident ray. Now this over here is coming out. So we call this the emergent ray. And then the ray inside here does go the refracted ray. So this is the refracted ray. Okay. So this is another normal. This line here is also another normal here. Now the angle between the normal and incident ray, that will be your I. So this is going to be your incident angle. Incident angle. And then the angle here, let's call it R. So this is going to be your angle of refraction. Refraction. Okay. And then if we take the angle here, we're going to call this the angle of emergence. Let's call it. So it's going to be similar to I. Okay. But let me just call it E. With the angle of emergence. It's coming out. Okay. So if you look at this, these are power lines. So if I have power lines, the normal here, the parallel to that here. So it means that the angle here is the same as the angle here. Okay, it's going to be the same. It's going to be the angle here is the same as the angle, alternate angles. So it is going to be the same as R on that side. Because of parallel lines. Okay. So that is refraction, the bending of light as it travels from one medium to another. Yeah. So light travels faster in air, slow in water, and slower still in glass, if you call the comparison. Right. The slower light is in the medium, the more it refracts or bends in it. Okay, so the more dense it is, the more the bending. That's what this means. Now, these are some measurements of what we call the refractive index or index of refraction. If you take air, the index of refraction is about 1.000293, water 1.33, glass 1.49, and so on, diamond. So you see that the ability to bend light is increasing as you move from like air, water, diamond, and glass to diamond. Okay, the degree of refraction. So there's something called Snelling's law. Snelling. If you see Snelling's law, there's a question about that. It asks which of the following instruments will you use to, in finding Snelling's law. Snelling's law is simply a measure of the refractive index. Okay. So refractive index. To measure that, you take the sine of the angle of incidence over sine of the angle of refraction. Okay, if you take out the sine in degree, so you measure the angles. If you measure that and divide by the sine of the angle of refraction, it gives you the refractive index, and that's how they got these values. Pass light through glass. You measure the angles that I spoke about in the lab. It gives you these values and find the sine of them in the ratio of the sines. So if you want to see if someone gives you a diamond, like a piece of substance, it says, oh, this is pure diamond. You can take it to the lab and check whether it's the correct one, whether the person is speaking the truth or not. To do that, you measure the refractive index. If you measure refractive index and it comes to 2.41, the standard, the one you know as pure diamond. If it's the same, then the person is giving you the correct diamond. If it's not the same, then the person is giving a fake one. Yeah. Okay. So that's why people are going to sell, let's say rings and other things. If you tell the person it's a genuine one, they take a light and they shine it in, or they put on those instruments. They are measuring the refractive index to see whether it's correct diamond or just a fake diamond. They're measuring the refractive index. All right, so the question that I asked was, what instrument will you use? In that particular question, they give ruler and they give protractor and some other things, So the answer will be protractor. You use a protractor. Since you have to measure angles, you need a protractor to do that, Okay. And then the last property is to know dispersion of light. Dispersion of light is simply the splitting of white light into its component colors. So you're splitting white light into its component colors. That's what we call dispersion of light. Dispersion of light. Okay. So if I shine white light through a glass prism, this in the lab use what called the triangular glass prism, you see that the rainbow colors come out. The colors are kind of like rainbow colors. You have R, Roy, G, Bive. Can use that mnemonic, Roy, G, Bive. Like Roy, G, assuming somebody's name. Red, orange, yellow, green, blue, indigo, and violet. Yeah. The rainbow colors. That's what you see. So if you combine the rainbow colors, you're going to get white light. You can go backwards. Yeah. So that's dispersion of light. So we talk about reflection that occurs at mirror surface. Dispersion, splitting white light into components. We talk about refraction, diffraction, the bending around small object of light around small objects. And then we also spoke about the refraction as bend of light as it passes from one medium to another medium of different densities. So those are properties of light. With that, that brings me to the end of today's lesson on the science review. Okay. Any questions before we go through the questions in the book? So now we're going to do questions in the book before, and then we'll do the math. Any questions? Okay, so I believe so far everything is clear. So let's see if we can apply all that we studied to some questions. So I'm going to go through the book and then I'll highlight questions for today. And if there's something that we've done already and I feel I have to bring it up, I'll bring it up. And if I notice something that I haven't touched on, and I may touch on it briefly. All right, let's go to page 218. Page 218. Number 1. 218, number 1. An organism with chloroplast in its cell is probably A, heterotrophs, B, autotroph, C, herbivore, and then D, primary consumer. So you can B, correct, autotroph, right? You make your own food, go to autotroph, correct. Page 219, 219. Number 7. As light passes obliquely from air to water, it is bent. This bending is called A, diffraction, B, reflection, C, refraction,
C.
and then D, dispersion. Yeah, C, refraction. So it's refraction of light. It's passing from air to water. The densities are different. So it's refraction, correct. So see, if you know the concept, you're able to move fast. Page 220, 220. Number 16. As the eardrum is made to vibrate more rapidly, the sound is perceived as A, loud in intensity, B, softer in intensity, C, higher in pitch, and D, lower in pitch. Vibrating more rapidly.
C.
C, correct. Higher in pitch. Rapid vibration, that means high frequency. You know, the number of cycles per second, very high. So it means pitch goes with frequency. So higher pitch, that means higher frequency, correct. So next time it could be with the sound, low intensity sound, high intensity sound. All right, so you have to also have that in mind. Intensity has to do with amplitude, frequency, pitch. Okay. See, page 227. Page 227. Number 52. In humans, if the diaphragm is pushed up, there's a decrease in chest volume. This decrease is followed by A, an increase in pressure in the chest cavity and inhalation. B, an increase in pressure in the chest cavity and exhalation. C, a decrease in pressure in the chest cavity and inhalation. And D, a decrease in the pressure in the chest cavity and exhalation.
B.
Okay, it's a B, right? So let's see. Let's analyze that. So if the diaphragm is pushed up, we may say that there's decrease in chest volume. So if there's a decrease in volume, what happens to the pressure? The pressure will go up, right? That's Bohr's law. So pressure will increase. So the pressure increases, what's going to happen? You have high pressure in the thoracic cavity than outside, than the atmospheric pressure. So it's going to push out the air. So we're going to exhale. So B is correct. Excellent. So that's Bohr's law right there. Okay. Page 237. 237. Number 31. It has to do with the diagram on the wavelengths, like waves, on waves. So it says the diagram shows 4 different waves traveling along the same path. Which 2 of the waves have the same wavelengths? They're the same wavelengths.
B and D, number C.
B and D. Try it again. Try that again.
Is it A and B?
Yes, A and B. See,
Why is it A and B?
say again.
Why is it A and B?
Now, the wavelength is a time for one complete cycle. So this is the wavelength here is for B. This is a wavelength for B. Complete cycle. Now, what is complete cycle? The time for one complete cycle for A. That's a complete cycle. Again, the same from here to there. They coincide. So you see the complete cycle brings to the complete cycle for B. If you take the other ones, it doesn't give you that. If you take D, D goes like this. It goes this way, and then it comes like that. You know, so it doesn't give you. It's not with D. It's not wavelength. You take C. C goes from here. It goes like, let me see. It goes like that. Comes through here, and then it's somewhere here like this. C. So it's only A and B have their complete cycles coinciding. So the wavelengths. Okay, page 239. Number 42. They say a ray of light travels from air to glass. So they give you the diagram air to glass. Which of these measuring instruments is needed to find the index of refraction of the glass? Then you have ruler A, protractor B, balance C, thermometer D. So I gave you the answer in my discussion. Yeah, you need a protractor. Okay. You have to measure angles. Okay, good. 43. Many chemical reactions occur more rapidly with platinum as catalyst. At the end of the reaction, the platinum is found to be A, increase in quantity, B, on change in weight, C, change into another state, D, combined into the final product.
B, on a change in weight.
Yeah, it remains unchanged at the end of the reaction, so it can be used and used again. Correct. Okay. Page 240. Question on the food web. Number 48 through 50. So based on the food web. 48. The correct order of the food chain represented in this diagram is, so they want to come out the correct order. You have grass, rabbit, snake, hawk. B, hawk, owl, rabbit, vegetable. C, grass, rodent, snake, hawk. And D, grass, rodent, rabbit, snake.
A.
You said A. Did you say A? Can you repeat what you said?
It's A, grace, rabbit, snake, hawk.
Grass, rabbit, snake, hawk. Okay, let's follow the arrows and see. So you have grass. Does the grass go to rabbit?
C.
Okay, see the grass goes to rodents, right? Then from rodent it goes to snake, and then from snake you can go to hawk. So C is correct. So you follow the arrows smoothly without breaking. Like you're traveling on the road without breaking to your destination. Trace it. 49, a necessary member of the ecosystem not represented in this food web is A, producer, B, primary consumer, C, decomposer, and then D, secondary consumer.
C.
C, so the decomposer is not shown here, even though it's an important member. Okay, 50. If vegetables were removed from this food web, which of these results would be the most probable? A, the snake population would increase. B, the rodent population would die out. C, the grass would increase. And D, the rabbit population would decrease. You're getting rid of vegetables.
B.
D.
Okay, somebody said D, so I said B. All right, let's see which is correct. Now, if you take out the vegetables, we notice that the rabbits will not have any other food because they only have vegetables as a source of food. So it means the rabbit population will go down. The rodents, even though they feed on vegetables, they also feed on grass. So if we take out vegetables, the rodents still have grass that they can feed on. So the rodent will still survive, all right? So the one that will die or the population will go down is that of the rabbits, because they don't have any other source of food than the vegetables. So D is correct. Okay. All right, another example here. 53. How much of 12 grams of radioactive substance with half-life of 20 years will be left after 40 years? So try that one and give me the answer. A, 0, B, 3, C, 6, and D, 8.
B, 3.
You have 3. Okay, after 20 years. All right, so let's see if you're right. Okay, so you have 12 grams, half-life of 20 years. So it means first 20 years, half of this will be left, so 6 grams. Another 20 years, half of this will be left, so 3 grams. We are dealing 40 years. So if you add this up, this is 40 years in all. So 3 grams, correct. So the answer is B. Okay. Look at page 248. Page 248. The food pyramid. Number 36 through 38. 248. You have the pyramid, they have green plants, insects, birds, and snakes in that order going up. So what first, if any, result would you expect if a disease decrease the population of the snakes? So something affecting the snakes. A, no change will occur. B, the number of green plants will increase. C, the number of insects will increase. The number of birds will increase.
D.
D, the number of birds will increase.
S. D.
All right, so the birds, the snakes feed on the birds, yeah, on top of the birds. So if you take out the snakes, then the bird population will increase. All right, so these questions, as I said, they're easy to answer. If you know that, you just have to follow the diagram. If you know the concept, you follow the diagram, that's all. All right, number 37. Which are herbivores? A, green plants. B, insects. C, birds. And then D, snakes.
B.
B, yeah. So the insects feed on the plants, herbivores. Okay. In the diagram, you can see they are the primary consumers. So those are the herbivores here. And then 38, according to this pyramid, to support 100 pounds of birds, which of the following is needed? A, more than 100 pounds of insects. B, more than 100 pounds of snakes. Less than 100 pounds and 100 pounds of insects. Yep. Somebody said A. That's correct. You need more than 100 pounds of insects to provide energy for them. Remember, it's a 10% rule. So those below should have more energy than those above. Okay. Now let's just add 39. Ozone is an allotrope of A, hydrogen, B, water, C, oxygen, and D, uranium.
C, oxygen.
Yeah, oxygen, correct. So allotrope. Allotrope is a substance that can exist in different forms in nature. Different forms. Oxygen can be in O2. It can also exist as an oxone, O3, in nature. That's oxone, the oxone layer. 3 oxygens combined. So it's another form of oxygen. Okay. Number 42. 42. It says a mixture such as mayonnaise is best described as a substance, a saturated, a substance, B, saturated, C, a solution, and D, emulsion.
D.
Yeah, D is an emulsion. Emulsion. Okay. Remember I spoke about emulsions. So emulsion. It's a stable one. Okay. Let's see. Okay, now 44. Now 44, you should be able to do that one. Use your knowledge from any physics class or science class. A woman standing at the bus stop hears the siren of an approaching ambulance. As the ambulance passes by her, she observes a shift in the frequency of the siren. The effect she heard is known as A, photoelectric effect, B, Doppler effect, C, phase shift effect, and D, Einstein effect. Make a good guess.
C.
It's that actually the answer is B. If someone said B, you are right. Doppler is called a Doppler effect. Doppler effect.
What's the Doppler effect?
That's what the question is describing. That's a Doppler effect. The shift in the frequency of the sound. It's an ambulance coming close to you. It appears the sound is approaching you, hear louder. If it's move away from you, it moves. it appears that it's moving away from you. That shift in the frequency of the sound is what we call the Doppler effect. "45. The pitch of a vibrating string depends on all the following except A, the length of the string, B, the amplitude of the vibration, C, the thickness of the string, and then D, the frequency of vibration." Make a good
C.
guess here. The thickness,
B.
all except, actually the answer is B. Okay, it depends on the, what do you call it? Or the length, the thickness, and then the frequency of vibration. Amplitude, you see, if you look at pitch, one we can look at this thing called the guitar strings and the strings of the guitar, right? If you vary the length, you have different frequency or pitch. If you vary the thickness, it gives a different frequency. Now, pitch is related to frequency. So you can use D as part of it. So pitch depends on the frequency. Now, amplitude is the odd one here. Amplitude has to do with the loudness. Okay. So you can do elimination based on the low knowledge that you know and everyday life examples too, if you're stuck. Okay, page 250. Number 47. Number 47. As the angle of incidence of a ray of light striking the mirror increases, the angle of reflection will A, increase, B, decrease, C, remain the same, and D, first increase and then remain constant. So you're increasing the angle of incidence. So what happens to the angle of reflection?
It increases.
It increases, correct. Remember we said that the angle of incidence is the same as the angle of reflection. So if you increase it, then that one should also increase to the same value. Okay, so increases, correct. Okay. Now 49, that one we know the answer. In a food chain, an organism that feeds on green plants is known as decomposer, producer, first order consumer, second order consumer. The one that feeds on the green plants.
C.
Yeah, it's a first order, primary consumer. Okay, primary consumer. Okay, so let me see, I have one or two more here. Page 252. 252. Okay. Number 50, no, number 60 is based on the diagram. Number 60.
B.
Yeah, which of the rays in this the best represent reflected ray? So B, correct. Okay, 30, 30 has to be 30, 30. So you choose B, correct. Okay. All right, so I believe at least you've seen some questions on how the topics that we've treated go. Okay. Look at page 290, maybe two more questions, two or three questions here, and then we'll break up, we'll stop. Page 290, number 4. Number 4. Say, which of these diagrams correctly shows a water wave passing through a small hole in a barrier? So this is an example of diffraction.
B.
B.
Yeah, B. Yeah,
B.
B, correct. You know, pass through the hole, spreads out, bends a little bit, the waves, you know, bend a little bit, so you see the spreading effect.
What was the problem page again? I'm sorry.
Or page 290, number 4.
290.
Yeah, 290, number 4. Okay. Okay, number 7. Let's look at number 7.
What was the answer for that one, for number 4?
It's B. Number 7. So they give you the formula, 2H2O2, then the arrow and the MnO2 above the arrow, and you have 2H2O plus oxygen. So the catalyst is A, H2O2, B, MnO2.
B. MnO2.
Yeah, MnO2. Yeah, it's above the arrow. That's the enzyme. That's the catalyst. Sorry, the catalyst. All right, number 8. The frequency of sound wave is directly related to its A, loudness, B, pitch, C, timbre, and then D, resonance.
B.
B, correct. Frequency goes to pitch. That's what I have to remember. All right, so yeah, I think you have to answer. Number 12, for example, is the same thing. The rib cage moves up, upward, what happens? Number 12, page 291. Okay, 291. Number 12. So when the rib cage moves upward and outward, the diaphragm flattens out. And say A, air is expelled from the lungs. B, air is drawn into the lungs. C, air pressure in the lungs rises. D, there's no change in the air pressure in the lungs.
A.
Yeah. So the diaphragm moves upward, rib cage moves outward, the diaphragm flattens. That means the volume has increased. So volume increased, pressure down. So pressure is down, it means that the atmospheric pressure is higher, so air will be pushed in. So air is drawn into the lungs. So B. Okay. Look at number 15. Number 15 is ecology. The first living thing found colonizing a hitherto barren area is called A, a producer, B, primary consumer, C, pioneer organism, and D, carnivore. You can do a good elimination and get the answer. The first living thing.
D.
First living thing. Did you say D or B?
The A.
Think about the key word in this question.
B. Primary.
Yeah. No. First living thing. First has to do with what? Think of the pioneer. Pioneer. A pioneer is somebody who does something first, right? Comes out something, the first person to detect something or invent something first, pioneer. So the first living thing found colonizing a hitherto barren area is called a pioneer organism. Pioneer, first to colonize the area. So pioneer means first to identify something or to be in a certain place. And you can do elimination. Producer, that's out. We know producer, primary consumer. We know out, carnivore. Again, feeding. So that cannot be the answers there. Okay. I saw one last question that I want you to look at, and then we'll pause. It was one half-life question. Okay, right. Then let's look. This is the last question, and then we'll stop here. Page 298. Page 298. Page 298. Number 52. Which process produces the energy in modern nuclear reactors? Which process produces the energy in modern nuclear reactors? A, fusion. B, alpha decay. C, fission. And then D, electron capture.
C.
Actually, it's C. C, nuclear fission. That's what they use a lot for generating nuclear, like modern reactors. They use that, generates a lot of energy for electricity and so on. Nuclear fission. All right, let's look at 54, will be the last one, and then we'll switch. Half-life question. The half-life of iodine 131 is 8.1 days. What fraction of the sample will be left? Or will remain unchanged after 32.4 days of decay? So take like a minute to think about that. So half-life is 8.1 days.
Is it B?
B. Okay, we'll try that and see if you get B. Did someone get anything different?
116.
1/16. All right, so you have two different answers here. Let's see which one is correct. All right, so first half-life 8.1 days. That means that half of this will be left, right? So 1/2 of the I will be left after first half-life. Okay, another 8.1 days, how many will be left? Half of this, which is equal to 1/4, right? Another half-life, 8.1 days. How many will be left? Half of this. So half of the 1/4, which is 1/8. Another 8.1 days. How do we have 1, 2, 3, 4? So we have 4 half-lives because we want up to 32.4 days. Okay. So that gives you 4 half-lives. So this takes you 1/2 of the 1/8. So that is 1/16 of the substance left. So the answer is D, 1/16 of the original will be left. Right. Right, so this is a good point to stop. So I believe you can answer the other questions when you go home. Next week will be a time to, we'll do a little test. First, we'll do the math and science, like 45 minutes. After that, we'll just do a discussion on the test and then answer any other questions that we may have. So we'll have more practice again next week on the questions.
Wait, Professor, I thought it was, isn't today the last?
So it says how many 2 1/4 fluid ounce bottles can be filled from a bottle that contains 9 fluid ounces. So what do you think we should do here?
D, number 4, 4 bottle, number D.
Okay, so how did you do it? So we all will get used to it.
I divided the 9 fluid ounces divided by 2/4 fluid ounce bottle.
Okay, so 9 divided by 2 1/4. Okay. All right, so this will help us review our fractions again, review of fractions. So we said 9/1 divided by the improper fraction, right, for this. So 4 times 2 is 8 plus 1. That would be 9/4. Okay. Since we're multiplying 2 fractions, we'll keep the first fraction, change to multiplication, and then reciprocal for the second fraction, 4/9. You cross out 1, 1, and then you have 4/1, which is 4. Correct. Okay, good. So number 2 is also fraction. You're adding. Okay. Good. So take like a minute or to do it, or less. Try not to spend more than 1 minute on a question. So now you should be timing yourself, right? When you're practicing, start timing yourself. You've studied enough, you have enough notes, so take your time to review and then time yourself when you're practicing questions so that you can gain speed. Timing is very important. Sometimes but if you don't time yourself, you can't be able to do a lot. Any answers?
Yes, number A.
A, okay. So when you have fractions, we said if you're adding, you need LCD, correct? LCD needed. So write each one with the LCD. What will be our LCD for this question? 4 and 6, 12, yep. So we have to ask ourselves, what was done to the 4 to get a 12? This is times 3. So we have to multiply this also by 3. So numerically, this will be 9. 6 times 2 gives you 12 in the denominator. So multiply the numerator also by 2. So that will be 10. Okay. Now, if you add the numerators, so this gives us 19 over 12. The whole numbers, you have 3. But 19 over 12 is an improper fraction. So you have to change to a mixed number. So divide the 12 into the 19. How many 12s do I have? I have 1 of that. 12, subtract, you have 7. So the 19 over 12 is the same as 1 whole number, this 1 whole number, 7 remain out of 12 left. So you're going to add a whole number to the 3. So the answer will be 4 whole numbers, 7 out of 12, 7 12s. So A is correct. Okay, correct. Okay. All right. So number 3, I believe we can skip that one. If you were to do that yourself. The subtraction. Number 4, I believe you should do that too, unless someone has a question. Number 3 and 4. Anybody has question 3 and 4? Should be fine. Okay. Number 5. Again, the division of fractions. I believe you should be fine with that too, since we've done one example, the first one.
A, 1/5.
Yeah, so that would be good. So again, keep the first fraction times reciprocal of second one is like over 1. So 1/4, so 1/5, correct. All right, so I believe you should have your fractions down packed. Dividing, subtraction, addition, and multiplication. Work on the fractions if you're still not too sure, do more of those things. Okay. Because the same thing comes the word problems, same application of the fractions. Okay, look at number 6. We can look at number 6. Tablet contains 2.25 grains of aspirin. If a patient takes 2 tablets every 4 hours, how many grains of aspirin will be taken in 24 hours?
27 grams.
27, correct. Okay. So 2.25 grains, and the person is taking 2 tabs every 4 hours. So let's multiply by 2. That means that the person will take, this would be 4.50 or 4.5 grains per dose, right? Every 4 hours. Now the question says 24 hours. So we're going to ask ourselves, within the 24 hours, how many 4 hourly sessions do you have or periods do you have here? You're going to have 6 of that, right? 6. So out of the 24, you divide by 4, you have 6 times. So you have to do 6 times. Each dose, you are given 4.5 grains. So it means over the 24 hours, you have to give 4.5 times 6. Okay. And then that's where it takes you to the 27 grains. Okay. Right, number 7. That's average. So average, you should be able to do that also easily. Okay. Everybody should be fine with number 7, right? You should be fine. Add all the numbers and divide by the number of weeks. First week, second week, third week. So 3. I saw 4. Divide by 4. So we should be able to do that one. Yep, 4 weeks here. Number 8, we should be able to do number 8, fractions, dividing fractions. So this question, and this test has a lot of dividing fractions here. So number 8 should be fine.
What's the answer to 7?
For number 7, add all the numbers 2.4 plus 3.5.
7 is going to be C, 2.95.
And then divide by 4.
It's C, 2.95.
2.95.
Oh, I forgot the 3.
Number 8, you should be able to do that because it's fractions, dividing. We've done a lot of this one.
8, the answer is D.
Okay, 5/3, which is 1. Five over 2. No, this is 6, sorry. I put 9 here. This is 6. So 3 here is 1, 3 here is 2. So it's 5/2, which is 2.5. So it's D. Okay, number 9. What is the maximum number of 1.5 inch strips of tape that can be cut from 480 inches?
C,
Of
320.
so 320. Okay. All right. So what did you do here?
I divide 480 inch divided by 1.5 inch strips.
Yes. So you have to divide. All right. So anytime they ask like you want pieces of something, you take the whole and divide into those small pieces. So at 480 the total length, and you want to break it down into 1.5 strips. So you have to divide, correct? So 480 divided by improper fraction, 3/2. Okay, you can put this over 1 if you don't make any mistake. Then keep the first fraction, change the second fraction into, find the reciprocal of the second fraction. So 2/3. And then you can reduce to lowest terms. Either use a calculator to multiply the numerator and denominator and divide, or you can reduce to lowest terms. 3 here is 1, 3 into that will be 160. So if you multiply 2 times 160, then you get 320. Okay. So C is correct. Okay, so look at number 10. The income tax system requires that persons having a net income between 10,000 and 16,000 pay a tax of 800 plus 24% of the part of income in excess of 10,000. How much tax should be paid on an income of 12,500? Okay. So it's 800 plus 24%.
B, 1400.
1,400. Okay, correct. You are correct.
Professor,
Yes. Go ahead, please.
oh, wait. I was just a little confused with how you got a number 9, how you got 320, because for some reason what I did here at 480 times 2/3, I multiplied 480 times 2. Is that
Yes. And then divide by 3. You should get the same answer.
Oh, and did I was okay.
Yeah, I like to reduce to lowest terms.
Okay.
It's so that's why I didn't multiply them directly.
Okay.
But if you use a calculator, then 2 times 480, so you get 960, and divide by 3, you should get 320.
Okay, thank you.
You're welcome. All right, so for number 10, this is a summary of what the question is asking you to do. 800 plus 24% of the portion that is excess of 10,000. Okay. So now the person gets an income of 12,500. What is the tax? Right? So it means that you have 800 plus 24%. You can work in decimals. So 0.24 times the excess of 10,000. If the person makes 12,500, take out the 10,000. So that will give you as the excess above the 10,000. Okay. And then if we work this out, we should get the answer, which is a 1,400. Okay. So 800 plus, then that would be what, 600, and I get $1,400. Okay. Number 11, we should be able to do that. Decimal, 3/7 to decimal. So you divide 3 by 7. If you have a calculator, punch in 3 first and divide by 7. Without calculator, that means you have to divide long division. 3 into 7 into 3, impossible, 0. Into this decimal point, add 0 to this, 7 into 30, then that will be 4 times. Then this will be 4 times 7 is 28. Subtract, that's 2 at 0. 7 into 20, that is 2 times, that which is 14. That is 6. You can keep adding zeros. 7 into that, that'll be 56. No. 49 times 8. So that is 7, that'll be what? Yeah, that's 8, right? $56, and then so 2 decimal places, so we can stop here. So the 8 is more than 1, more than 5. So you're going to round up. You round the 2 up to 3. So 0.43. So the answer will be C. Right, you can use a calculator to get the answer for this, since you're allowed to use it. Okay, number 12 says, what is a prime number that lies between 95 and 100? Prime number. So they're testing you if you know prime numbers. So a prime number is a number that has only 2 factors, one in itself. So which one would be the prime factor between 95 and 100 here?
97.
I didn't hear you.
B.
B, correct. Yeah, 97. B. Right. We can only have 97 times 1, nothing else. So that's a prime number. Okay. Number 13. Three-ounce serving of corned beef hash supplies 155 calories. So 3 ounces gives you 155 calories. So approximately how many calories will be supplied by 5-ounce serving? So 1 5-ounce serving. What is the calories? So that's a summary of the question.
Is it D?
You had D. B. Yes, it should be B. Okay, you see, like, again, we can, you see, D is too small. If you look at 3 ounces giving you 155, and in 5 ounces, you expect like more, a little bigger number than a 155. Okay. So here you can use proportion. 3 ounces gives you 155 calories. Set out the same ratio, ounces and calories. Yeah. You've been given 5 ounces. So the 5 ounces, that'll be 5 here. You don't know this part here, X is missing. So that's a proportion question. And if you do your cross product, 3 times X is equal to 5 times 155. And then divide by 3. Divide by 3. And then you get the answer. So that would be 5 times 155. Let's check that and see. Let's pause. 5 times 155. So that's 775 divided by 3. We get 258. 258.33333. So the answer here will be B, 258, to the nearest whole number. So B. Okay. Okay. Number 14. Number 14. So a jogger travels X miles each morning. Which of these equations represents the number of days needed to jog 200 miles? So each morning, X miles. How many days to jog 200 miles? So the first one, 2x, 20x, then you have B, 200x, then B, 200 over x, C, x over 200, and then D, 200 plus x. Which formula is correct? Or which equation is correct?
B.
B, correct. You have to divide the total distance by the distance traveled each year. Okay, so it's 200 over x. B, correct. Number 15, you should be able to do that easily. Negative, negative. So that shouldn't be a problem because opposite of a positive, negative, negative, positive. So 176 plus 64. Okay, that's what we want to do. So 4 plus 6, 64, 4 plus that's 0. 1 left, 1 plus, that's 714. So 240. Okay. Number 16, exponents. Multiplying exponents. We went over that too. So how should we multiply this one?
Multiply the bottom, add the exponents.
Yeah, we're going to add the exponents. So multiply the numbers 3 times 2, which is 6. Since you're multiplying, we add exponent 2 plus 4. So that would be 6. Correct. So it means the answer will be B. Okay. Number 17. We are factoring. Okay, so this is what we call a difference of two squares. Difference of two perfect squares. All right, so if you are factoring difference of two squares, it's always in the form a squared minus b squared. Okay. We take the square roots of each number. So a squared root is a. So put one in each bracket, parentheses, a times a. And then take square root of b, which is b, one in each parentheses. Then you add and then subtract the two roots, the difference of two squares. So you have 49 a squared minus 49. And you want to factor this out. So here, just put factor here so you can remember factor. So to factor this out, you know, if you look at this, these are perfect squares, right? For every factoring, the first thing you do is to look at common factor. Take note of that. You need a common factor first. So if I look through, there's no common factor, right, to factor out. But then I know that it's special, they're perfect squares. So since they're perfect squares, I can break it down into the square roots. 4a squared. If I take the square root of this, this will be what? It's 2a times 2a. So the square root is 2a. So put 2a, 2a. 49, perfect square made up of 7 times 7. So the square root of that is 7. So 7 and 7. So add and subtract. Or you can subtract and add. It doesn't matter where you put a sign. It could be either in either parentheses. Okay. Okay, so the answer for this question is going to be D. Factoring difference of two perfect squares. Okay. All right, number 18. I believe that should also be straightforward for you. It says a man estimates that 1/5 of the salary is spent on taxes, 1/4 for rent, and 1/10 for insurance. What fraction of the salary is left for other expenses? Left. So if somebody wants to take us through the approach. So salary, one-fifth of the pie. Rent is 1/4 of the pie. And the insurance forms 1/10 of the pie. So the question is how much is left for other things here. So if you're dealing with fractions, remember the whole thing is one whole number, all right? One whole. So if I add all these fractions, I'll get the amount spent on the salary, rent, and insurance. So let's do that. So 1/5 plus 1/4 plus 1/10. I need LCD.
20.
20. 20, 20. So this is times 4, right? So this will be 4. Times 5 to get 20. So this is times 5. So 5. Times 2. So times 2. So what do we get here? 4 plus 5 is 9. 9 plus 2 is 11. So we have 11 over 20, right? That's the sum of all this. So the piece that's left here, I have to take it out of one whole. The whole thing is one whole. So 1 minus 11 over 20. Okay, so what should I get? 20 parts, I take 11 parts. You fuse 11 parts. So how many parts left? Yes,
9.
9. Yeah, so 9 over 20 left. Okay, so you take 11 out of 20 and you have the portion left. Or you can 1 minus that, which is the same as 20 over 20 minus 11. That's what that means, which is 9 over 20. So A. So this section has a lot of fractions, here. Number 19. If you look at number 19, we'll run up shortly. Let's,
Professor, you subtracted by 1.
yeah, we subtract from 1. Because the whole piece is one, like 1 pi, which is 1 whole. You see, when you do fractions, you take the whole piece is 1. When you take the whole piece. So if you use 11 out of 20, you know, the parts is 20 parts, right? You use 11 out of the 20 parts. So the portion left will be 9 parts. 20 minus 11, so 9 parts left out of the 20. That's the 1 minus 11 over 20. That's what that means.
Okay.
Okay. Okay, number 19. Let's see if we can round up on these ones here. Number 19, you have to 2 over 3 of x is equal to 48. Then they say, what's a half of x?
C, 36.
C, 36, okay. Right. So you can change this to algebra first, right? You write in algebra form, so it's like 2 over 3 of means multiplication. So 2 over 3 x is equal to 48. Since 1x, you multiply the left side by the reciprocal. You can multiply by reciprocal of the fraction, so this 3 over 2. Whatever you do left side, just same to the right side. So this will take care of all this. They have just x. Then we can simplify this end here. So we have 48 over 1 times 3 over 2. So I can reduce to lowest terms. 2 here is 1, 2 into 48, that is 24. So that gives us 3 times 24, that was 72. And the question says 1/2 of the x. So it means 1/2 of 72. X is now 72. And then that gives you 36, as she said. So 36C, correct. Okay. All right, so number 20, let's run up to 24 and then we'll stop. Just in like 3 minutes. Number 20, that one you want negative of 2 raised to the power 4, right?
G, minus 16.
Yeah, minus 16. Minus 16. The negative sign is outside, so it stays. So outside, so it's negative 16, correct. See, this will be different if you have something like negative. If I write this, then that will be different. Okay, this one is negative 2 times negative 2 times negative 2. So I expect this one to be positive, but an even number of negatives, that's what that means. So that gives a positive 16. So that's different from this one. The next one is outside, so we maintain it. Okay. What are 21? Now it says 33% of X is equal to 99. What is X?
D, 300.
Yeah. So here, if you're stuck, you can work backwards, right? You can just take the numbers and then work backward, find 33% of each number. And then you should get 99, which is 300. You can work backwards. Or you can use your algebra to do this. You can set a percent proportion if you're stuck. You can say 33% is given off. You don't know the off, which is the base. So X, and then it's 99. This is equal to means is. So 99 represents 33. What is the base? Okay. And then if you do this, it will give you the 300. Yeah. But as I said, you can easily work backwards here. Take a calculator, 0.33 of the number. And if you look at this, the biggest is 300, so it's obvious. Okay. Number 22, another percent problem. So you can use percent proportion if you're stuck. So here off 200. So off is a base. Is 250. 250 will be the part. So is over off, part over whole. You want a percent, you don't know X, but the 100 always goes to the base because in terms of 100 is a whole. So that would be your percent proportion. Okay. And do the cross product. 200X is equal to 250 times 100. Excuse me. Then you can divide by 200. Excuse me. 200, so we cross out the zeros. So we have 250 divided by 2. And then that should give us 125%. Okay. So that is a percent proportion approach. Then what about 23? 6 less than X less than or equal to 7. They said X is an integer. If X is an integer, then the solution set of this 6 less than X less than 7 is A, 6 and 7, the set, and then B, set 7, and then C, set 6, and then D, empty set. So which one will be correct? If you take 6 and B, correct, 7. All right, so 7 lies between, is less than or equal to, so 7 equal, X is less than equal, so 7 is equal. So which is correct? If we take the first one, it's not correct because 6, the number 6, if you take 6, 6 is not less than the 6. So it cannot be correct, right? But if you take 7, 7 is equal to this 7. So that's correct. So B is correct, the set 7. It's correct. Then the last one, 24.
C, 130.
Correct. So that's a parallelogram, right? So opposite sides, opposite angles are congruent or the same. So this 50-50. You don't know this. This is opposite angles are the same. So this should be X and X here. Okay. And we know that if you add all this, you get 360 degrees. Okay. Or if you focus on one side, 50 plus X, you go to 180. This 180 on this side. you can use that approach. Okay, or you can just say 360, no. So 50 plus 50, so x plus 50 plus 50 should give you 360. All right, so 2x plus 100 is your 360. Subtract 100 from that. So that would be 360 minus 100, which gives you 260. Divide by 2, so your x will be 130 degrees. In other words, add the 250s, which is 100, subtract from 360, and whatever you get, split it into 2 parts, divide by 2 to get it. That's what the algebra means. Okay. All right, so I believe it's a good point to stop for today. So please try and do more problems time yourself. As I said, next week we'll take we'll do the practice next week, and then we'll answer. We'll go through all the questions, the ones that you're not conversant with. You can ask questions on those particular questions, and then once we finish that, we'll go back to the textbook and to see other problems as well. Okay, so next week is practice time. Okay, any questions before we leave for the day?
Thank you.